Good evening, class. Welcome to the 8th session of a Proteomics. I hope you are all doing well. Had a good day, whole morning and afternoon, and looking forward to the sessions. So, and if you have any questions regarding career or any subject things, please feel free to ask now. Because we were doing yesterday blotting techniques. So within the blotting techniques, I forgot to make you done gel electrophoresis. So today we will be dealing with the one um, what is this virtual lab of gel electrophoresis. Yeah. So there are softwares which you can check how much you are copying. So there are some free softwares available. Plagiarism software checker. So these are the various plagiarism software checker. Viper is there, small SE tools, Grammarly. So all these are free checkers which can tell you how much it is there been copied. So it should be should not be copied from any paper. So okay, this is your first time. So okay, I agree. I mean, mostly it's agreed 5% or so, but okay, I agree to some 20% for you. So you can copy to some extent, but try to make your words in your own words, everything. Try to have a new words from that paper. Try to make new sentence to get a summary out of that paper. The best way to do so, to check the to write a paper, is you first read at least 15 to 20 review papers. Yeah, you read at least 20 review papers, then you start to make your own story. Just start writing your things in your own words, and then start giving the references later on with the context that you're seeing. I mean, you will publish in free journal, so it's not gonna be much. Yeah. So I mean, below 50% is acceptable by the generals. So if it is more than 25%, then it's not acceptable. So 15% is acceptable still. But if it is 25% or more than that, then it is not acceptable. So best review general is to look for like annual reviews. Go to PubMed. So some review will come. Molecular pathology of cancer, from pan genomics to post genomics. So like this, uh, you will have lot of journals on PubMed in huge amount. They are 120 around so far. So just read these abstracts and then go for it. Then try to get your own story out of them. So there are more impact of proteomics on anti-mycobacterium tuberculosis (MTB) vaccine development. Just click one journal. So this is free journals that you have to check. So I'm working from home at the moment, so I cannot access them. Polish Journal of Microbiology. These are some Polish journals from Poland. In this way you can do it. So 15% is allowed, but if it is 25%, then it's not good. It's bad. Yeah, I mean, a review paper will be enough to write a review, because this will be a lot. Because in the review paper, you already have summary of everything, all the research papers. So I will not. I will. You can read it for your own knowledge, but reading at least 10 reviews will be enough to write one review out of them. So that's the best policy to do so. That's all the big scientists do also. They write some references of the journals also, of which they are interested, but mostly review paper will help you to do so. Yeah. So I already have shared with you free journals, right? Anaconda, I don't know, bache. I mean, Pymol. I mean, I can discuss about this soon, about Pymol also during my PhD thesis. Maybe one day I can share my PhD thesis. So where I will share with this. Before I go ahead with the Pymol thingy, I want to finish with the blot techniques. With blotting technique, we did this gel electrophoresis. So there is some very nice virtual lab. First, we go through it. So we have a DNA with all the sizes, short, large, medium, in our mixture. So DNA strands are molecules, so we can't see under our microscope or our naked eye. So we need to see them via gel electrophoresis. So scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is useful for separating the other types of molecules like proteins and how they work. So this is your gel. So gel, this is the gel where filter. You can see some pores, my molecular sieve tubes. So you put your samples on top, and we place DNA sample into holes at one end of the gel. An electrophoresis is done, and we can see from negative to positive your current is moving out, and your DNA will move from negative to positive because of the phosphate group, because DNA is negatively charged. So your DNA moving from the sieve like this, Shorter will move faster, medium will be in the middle, and longer will be at the top. So staining the sorted group of DNA makes them visible to naked eye. Although we can't see a single DNA strand, we can see large group of stained DNA strands. So these groups show up bands in the gel. So we can see some bands here. So let's start the gel electrophoresis. So first we make the gel, then we make this a gel apparatus, load the DNA sample into the gel, then hook up the electrical current and run the gel, and then stain the gel and analyze the results. Welcome to the gel electrophoresis laboratory. So here we need agarose, flask, gel mold, gel comb, buffers, microwave. So all these things are required. So we have seaweed agarose. We took one spoon of it and put into the conical flask. Then add some liquid buffer to the flask. The buffer is a salt solution which has electrical charges. Then you put into the microwave. Let it boil. It's in liquid form now at the moment. Now we pour into the molds. Now it's in liquid state, so we can pour our comb to make holes into this. So an hour, about half an hour, it will be enough to make it solidify, and then comb could be removed. So we have holes in our gel. So now you put electrophoresis box into that. Put some buffer which has charged particles, and then you put your chamber into that. So you have loading buffer, DNA sample, DNA size standard, gel box with buffer gel, pipette box, micropipette. So loading buffer, we use the micropipette to suck up the loading buffer and add DNA sample. And clears liquid, because DNA looks clear liquid, so we added this blue color to make it visible with our naked eye. And this makes DNA sample thicker, so it will not drop into instead of floating away. And DNAs are standard already contain loading buffer, so that's why we are adding this loading buffer to it. So it's blue in color. You add into that DNA sample. So next, use the micropipette to transfer the DNA sample from the tube into the well of the gel, and then first suck up the DNA sample. So let's have the same DNA sample now with the loading buffer, and we pour into the well. Do it carefully so that it should not come out, it should go inside of your work. To the second one, you add DNA science standard, that is molecular standards, so that we can know that how much size our sample that we are running is. Like this. So now it will be attached with electricity, black with the black, red with the red, and we turn it on. So it will start moving, bubbles will start to come. That means that it started to run, and your sample will start going downwards. Over the time, shorter DNA will be migrate faster than the longer ones, and we can see them via blue dye also. So now it's time to analyze the data. We put them into the ethidium bromide, which will attach to the base pairs of a DNA. Like this. It takes about half an hour to stay in the gel, and we will speed up clock for us. And then we check under the UV light our DNA. Turn on. So here, so how much size are these are? So this is the molecular ladder that we have added, DNA size standard, and on the left, how much do you think these sizes are? So how much on the first one? What do you recommend? Just think about it in your mind. How much will be this one? How much will be this one? How much will be this one? So to me it seems around 150, 1500, and this seems 3500, and this seems 6000 base pairs. If it is so, then we click yes, forward. It will tell us. So all of the estimations are correct. So the top band pair is approximately 6000 base pairs, second is 3500, and the third one is in the 1500. So we have just run our gel, and so that's it. That's how from the start till end you run your electrophoresis gel. But the last paper submission is 25th of October now. Earlier it was 20th October because students need more time to submit. So you can submit it by 25th of October. So we are done with this gel electrophoresis. Now let's start with today's technique. Fluorescence activated cell sorting, FACS analysis. So that's the main motive of today's session. How to do fluorescence activated cell sorting. So basically in this your cells are sorted out based on the charge on them, on the type of proteins you have, and then you sort them into the forward and side scattered light. Then the detection at the forward scattered and side scattered, according to that we can differentiate between your cells. If they are forward scattered, that means the cells are big cells. The bigger the cells, the more you see the cells on the forward scattered. The more cells you see on the side scattered, they are the more complex ones. So that's the basic principle behind fluorescence activated cell sorting. To understand better, let's start today's lecture. In this lecture, we will be dealing with the basics of flow cytometry, understanding how flow cytometry sorter works, understand how cells are sorted out, description of different cell sorting possibilities, tips on sample preparations, and overview of services provided by FCF. So there could be two types of samples you can have while preparing your cell appropriate for cell separation. So there could be fluid particles in suspension and tissue ones. The one which are in the fluid suspension, they could go for three different parts. They could go for FACS, DAP array, and COPAS. whereas in the tissue one you can go for laser dissection microscopy. But today's interest is in our FACS analysis. So what is FACS analysis in this? Flow cytometry means flow means fluid, cyto means cells, metry means measurement. So you're measuring the cells which are in the fluid, which are in the flow of moving. So what type of particles can be analyzed? So particles that could be analyzed are cells, chromosomes, bacteria, yeast, microparticles. So all these things are measurable in the field of flow cytometry. And what kind of sizes of cells you can measure? We can measure from 0.5 micrometer to 14 micrometer. So including phytoplankton, red blood cells, CBA beads, lymphocytes, neutrophils, monocytes, etc. So what does it measure? So the key parameters are measured in this: a relative size, a relative internal complexity, a relative fluorescence intensity. So what is how is the size of a cell is, how the internal complexity is, how is the relative fluorescence intensity is. So all these three matters are measured during your FACS analysis. So these are various analyzers and sorters by BD Bioscience that they look like. So these are the various according to the complexity they are designed. A sorter can be divided, the one that is sorting could be divided into three parts, that is fluidics, optics and electronics. The fluidic parts will deal with the introduction of a focus cells, interrogating of it and creating a stable break-off for sorting. Optics is to generate and collect the light signals, where the electronics to convert the optical signal to proportional digital signals, processing the signals and communicate with the computer. So dealing with the computer part, electronics, optic dealing with the light signals, fluidics dealing with the cell part. So how we record them? So first the sample is introduced into the running of sheath fluid into the flow cells, right? The sample is hydrodynamically focused in the core stream. So you have samples in the center. On the side we have sheath fluid. So we accelerate them, leading to the laminar fluidics. Sample flow rate could be adjustable, and sheath fluid and central core cells, they are not mixed together. They always remain separated. So by putting them in this narrow structure, we increase the speed of our cells into single cells sorter. So they could be now 10 microliter per minute to 60 microliter per minute from low to high. So there are two different types of optic systems in a sorter, that is lasers and fiber optics cables. So fiber optic cables that carry beams to steering prisms, which then direct laser beams to the stream. So these are fiber optics, and these are the X mount optical plates. So you put them beam and using the prism, and then they go through your flow cell size through the nozzles. So like this, your various fluorescence light, they travel through the prism and they got condensed together and they are going through our flow cells like this. Then there is a collecting in the optics. Collection optic consists of fiber optics and PMTs, that is photon multiplier tube. Then your filters could be arranged into trigon or octagon shape. Trigon could be like A, B, C, and this could be like A octagon 8, A, B, C, D, E, F, G, like in opposite direction. G, H, A, B, C, D, like in opposite direction. Then photomultiplier tube is used to have a cathode of electron beam. From cathode we have a photon of light which goes through a lot of diodes, and from these diodes it got multiplied, its energy is increased to be absorbed at anode. So how cells are actually sorted out? So here this is the method actually. So first sample generate light scattered and fluorescent signals are analyzed. So samples are generated. Then second is charge is applied via stream charging wire. Then charge droplets break off. Then deflection plates attracts or repel charged droplets. Then uncharged droplet pass to the waste. Then the sixth step, so these are negative and positive sides. So these cells become positive and negative. So they get attracted. In the center there is a neutral cell. So these are the waste material where things goes wasted it out. So then you can sort them into red and green part, and interest of cells are sorted like this. So that's the basic principle of FACS analysis. So this is on the left side is interrogation point. So here you can see first uh, there's a direct droplet of your FACS analysis. Then there's a droplet started to happen, and then your sorting is starting later on when the discharge tubes are present. So live image of the stream taken from the video monitor. Drops are visible because the stream is illuminated by the stroboscope lamp, which is driven by the drop drive frequency. So what collection devices can I sort my samples? So you can sort your samples into bulk sorts. In the FACS tube, 15 milliml tube, beakers, sample tubes could be used. You can also sort them in the plate sorter with 6 to 384 well plates. So you can also sort them there. You can have slide sorters. In the slides, you can sort them with the help of microscopy slides or LBD type chamber types. So quick tips on sample preparation. Plan experiment well. Choose your fluorescent antibodies in agreement with optical configuration of intent sorter. So unstained or estimate background signals. Bring single strain sample for the compensations. Maximum of 2% of FCS has to be used while you're doing experiments so that cells are not sticky. Avoid phenol red media because of autofluorescence. It's also tendency to use calcium magnesium free sorting buffer or add EDTA to that. Use DNase and magnesium chloride, or also use 1% aqueous in sorting buffer. So these are the various measures you can do while you measuring your samples. So all samples need to be filtered, preferably different before sorting. So it's need to be filtered in the 70 micrometer, 85, 100 micrometer if your cells are 40, 20 and 10 into 10⁶ cells per ml. So avoid using mercaptanol triazole in these techniques, so that you should not be having any contaminations in your samples. So in summary, we can measure the analyzer, we can sort our sorters, prepare the samples as per the guidelines, do not use forget to controls, bring appropriate collection media for your downstream techniques. So thank you on the behalf of Edufabrica team. Thank you so much for being there. So now I will take you through some very nice videos. First I will share these things with you so that we should not forget. I share everything at the moment. So this is, we are done. So any questions so far, students? Internship certificates you get after the review submission. So you submit your top with your topic to us, then you can get your certificates later on. Yes, yes. So Western blotting technique has been used in the forensic field also to check between the crime material place and crime material and the suspected person. So you can check the protein samples from the blood samples and then run via gel electrophoresis, and then you can say that okay, this person's, if the same coming out, then you can say that the person is being culprit and has been not done the things as it should be. So yes, it is. So now I take you to some videos. So sit down, enjoy some videos now. So they will tell you, explain about forward scattered and side scattered, how you make a 2D histogram table, and how you make how to analyze your data from the blood samples. That's the main idea behind this. So I will start.
Guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually, I did two videos, and in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell passes through the laser beam, there are two events happening. First is the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC or a data analysis analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now I'm going to speak more in details. Saying that this is the flow cytometer tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak like high voltage peak for large cells and low voltage peaks for small cells. The data or the forward scattered light data at the end will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction. Yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of light into voltage, and then I will get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I'll speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch the next video. If you like this video, please don't forget to like, share, subscribe to the channel, and if you have any questions, leave it in the comments. I will answer you, and see you in the next video. Bye.
So students, you understand how you differentiate between your monocytes, neutrophils, eosinophils, basophils, having a forward scatter and light scatter, having the complex and the large cells on top, and simple cells and small cells to be here like debris, and big cells and simple cells to be on this side, complex cells and small cells to be on this side. So in this way we can differentiate between our cells. Now comes the next part, FACS analysis. So that was flow cytometry before. Now comes the next part of it.
Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer, and in this video, I'm going to speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I'm not like I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now when the cell passes through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore passes through the laser beam, the fluorophore will be excited. And then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scattered light. When the cell appears here, it means that these cells are small cells because they have low signal on the forward scatter and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appear here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scattered light. Then when the cell appear here, it means that it's expressing the protein, but it's a small cell. And when the cell appear on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescence cell with positive charge, for example, and the low fluorescence cells with negative charge. I can choose actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescence cells with a positive charge, and the big low fluorescent cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them. So it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... Of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelength. Then if the cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with the protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FACS. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector or on FACS detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about data analysis and gating in FACS analysis, please write me in the comments. As you know, now we can in the new FACS machines, we can analyze like tens of proteins and tens of cells in one time, because new FACS machines have like some machines like there is now one FACS machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel. If you have any questions or any suggestions for other topics, write me in the comments, and see you in the next video. Bye bye.
So that was quite in details about FACS analysis. In the end, it's all about the data analysis, that how you could interpret your data from the two proteins data here. In the earlier case, we have forward and side scattered. In this, we have two fluorescent dyed proteins. So according to that, we have different data. So any questions, students, so far? So I will play one more. This one, this one animation for all of you.
For more information on validated products and optimized,
So that's it. So everything is shared already. So students,
intracellular flows.
so you can take a 5 minutes break, or shall we finish it off with the virtual lab of flow cytometry? So it will not take 10 minutes or so. So we can start with this also then. Yeah, everyone is with me, awake, all good. Please thumbs up if everything is fine. Did you understand what has been explained today? With flow cytometry, with the forward and side scattered, with the fluorescence data, fluorescence 1 and fluorescence 2, how they are moving with the shear sheath and everything. Okay, good. So give me your some comments so that we know that things are going fine.
And multiple types of blood cells, each of which carries out a. Select a blood cell to learn more.
This is about our blood cells.
When we have a cut, platelets help stop the bleeding by allowing blood to form a clot. Red blood cells carry oxygen from our lungs to all of the body's tissues, and they carry carbon dioxide away. White blood cells help us to fight infections. There are multiple types of white blood cells, each with a different specialty. In a healthy person, each type of blood cell is present in predictable proportions. If the proportions fall outside of what is considered normal, it can be a sign that something is wrong. A complete blood count, or CBC, is one of the most commonly ordered lab tests. The test measures the proportions of different types of cells. CBC tests used to be done manually by looking at blood through a microscope. In a few minutes, a technician can count about 100 to 200 cells. Let's see how many you can count in a minute. An automated blood analyzer, on the other hand, can count many thousands of cells in just a few seconds. Its speed and accuracy make it a much more popular choice. The machine draws in a small amount of blood. Red blood cells and platelets have distinctive characteristics, so they can be counted with a method based on electricity. The differences between white blood cells are more subtle. To tell them apart, the analyzer isolates them and then uses flow cytometry to count them. The cells pass through a narrow tube in single file. One at a time, they pass by a laser. When the light hits the cell, it scatters. Across from the laser, a detector senses the amount of forward light scatter. Larger cells cause more forward scatter, and smaller cells cause less. Another detector senses the amount of side light scatter. Cells with a lot of internal complexity cause more side scatter, and simpler cells cause less. Computer software counts the cells and plots their light scattering properties on a graph. Since each cell type has a different scatter profile, we can use the graph to identify the cell types present in the blood sample and their proportions. Does this person's blood count look normal? Compare these results with those from a healthy person and note the differences. The plot doesn't look quite right. This person has many more immature white blood cells in their blood than normal. To get a better sense of what's going on, take a look at the blood under the microscope. The results suggest acute leukemia, a type of blood cancer. The patient's doctor will follow up to see if these results line up with any symptoms the patient may be experiencing. Flow cytometry is a tool that can be used to get information about your blood. In this virtual lab, you will use flow cytometry to do a complete blood count, diagnose a type of leukemia, a blood cancer, and look for any signs of cancer that remain after treatment. Leukemia happens when a stem cell in the bone marrow starts to divide out of control. Bone marrow is where most of the cells in our blood come from. In a healthy person, stem cells there divide to make new cells, which then mature and move into the blood. In the acute type of leukemia, immature white blood cells grow too quickly and fail to mature. They soon crowd out the other cell types. To stop the leukemia cells from growing, it is important to begin treatment right away. But the right treatment depends on identifying which type of cell is growing out of control. The CBC results from our patient suggest that they could have either B-cell or T-cell leukemia, but we don't know which. These cell types look the same under a microscope, yet they respond very differently to treatments. To find out which cell type is growing abnormally in our patient, we will need to analyze a sample of their bone marrow. Add some gene, but the tags will allow them to be differentiated. The blue tag attaches to a molecule on the surface of B cells. The orange tag attaches to a molecule on the surface of T cells.
Sorry students, some errors with the internet, with networking issue. Let me go through the last slide then. So you are basically understanding how we are differentiating between B cells and T cells by adding different tags, and then we run under the flow cytometer and we can differentiate between these two cell types. That was the main, and we can say that whether it was happening because
Flow
of B cells or
cytometry
T
is a
cells.
tool that can be used to get information about your blood. In this virtual lab, you will use flow cytometry to do a complete blood count, diagnose a type of leukemia, a blood cancer, and look for any signs of cancer that remain after treatment. After 4 weeks of treatment, flow cytometry is used once again to look for cancer cells that remain in the bone marrow. This testing is important because if cancer cells are still detectable, we'll want to give the patient a more intensive therapy. If there are no detectable cancer cells, then we know the treatment is working, and we will continue with it. Flow cytometry can analyze many cells very quickly, and it can detect even one abnormal cell among 10,000 healthy ones. This makes it the most sensitive and accurate measure available. It looks like treatment for our patient is going well. Back in the 1960s, B-cell ALL was deadly about 95% of the time. But today, thanks to flow cytometry and improvements in cancer care, 95% of patients with this diagnosis will recover.
So for first timers it will be complicated, but all in all, that's it for today students. I will share these slides also, go through them later. So any questions, anything else? Suggestions? So this is very complicated technique, so that's why I took one whole day to dedicate it to this, so that you are, whenever you have got a chance to work in the proteomics department, you got FACS analysis to work with, you can work there with this technique. Okay student, then good night, wish you a good sleep, take care, stay healthy, wake up early in the morning, do many things as possible, and we see us tomorrow at the same time, same place. And something about, maybe we study about bioinformatics tomorrow, so something related with bioinformatics, one small class about it, how bioinformatics is playing big role in the moment in the world, how we can make things out of it, so something related to that. Okay, students, thank you very much. Yes, yes, why not? Proteome bioinformatics, really exciting. Looking forward to it. It seems quite good, some innovative ideas. Proteome bioinformatics. If you can write, then go ahead. Okay, good night. Nighty nighty.